Thank you for introduction. I'm happy to be here. So I'm really interested in the morphological aspect of the organisms. So then I developed the theories and the techniques to quantify the morphological property of them. So today I'd like to talk about the morphological diversity and the several constraints on the leaf venation patterns. So I expect all of you guys have seen the leaf venation, leaf vein structure like that. So then the leaf venation of the angiosperm, especially the dicot, exhibit the hierarchical, the reticulate, the structures. So, then the first to third relatively thick veins called the major vein, and the fourth to the higher order relatively thin veins called the minor veins. So, and these leaf venations are also considered as the very important characteristics for the taxonomic classification, and some clades showing some specific venation architecture. So, and the leaf venation it is the transportation network of the water, the nutrient, and the chemical signals, right? So then the people believe that the leaf venation structures contribute to the various type of the physiological and the ecological functions. So, for example, as I say, the leaf venation is the transportation network. So then the hydraulic conductivity through the leaf it strongly correlate to the leaf venation structure. So, for example, the vein length per unit area (VLA), as known as the leaf vein density, is known as correlate with the leaf hydraulic conductance. So, in addition to that, under the natural condition, the leaves are often damaged by the physical perturbations and heavy loss. So then, some researchers reported that this venation pattern contribute to the tolerance of the damage. Then it means that if they have some passes, alternative passes behind the damaged region, it's beneficial to the survivorship of them. So in other words, how can I say, if the venation pattern have the detour, it's good. So then I believe that this venation pattern should be quantified as a network. So, however, most studies evaluated the leaf venation patterns based on the relatively simple measurements, like the length or diameter or branching angle of the main major veins. And as I say, the VLA or vein density. And sometimes it's also used the areole, which is the smallest area of the enclosed by veins. It is used for the index of the degree of the loopiness of the venation patterns. So is it enough? Are they enough for characterizing the whole venation patterns? So I believe that we need another aspect of that. So then, recently the topological approaches have been proposed. So I think that this is the promising direction because the vein structure is intrinsically represented as a network. So then in this study, we firstly developed a simple high-throughput phenotyping framework for quantifying the vein network based on the specimens. And then by using this method, we investigated the morphological diversity of several constraints of the leaf venation network. And finally, we identified the latent space of the leaf venation pattern. So firstly, I'd like to explain the framework of the purpose in this study. So in this framework, we assume the two types of the data as an input. So one, the untreated image data, leaf image data, is just digital data, digital images of the leaf data. And the other one is the cleared leaf data. So traditionally, the cleared leaf data are used for the observation of the anatomical detail, anatomical structure of the leaves, including the venation and the vein structure. So based on these image data, we firstly segment the leaf venation area by using a segmentation model, then extract the graph representation of the venation pattern. Then finally, the network features are calculated from this undirected graph. So then it means that we can represent the one venation pattern on the leaf can be represented as one vector, right? Then, yeah, we can enjoy on the vector data any types of the statistical method. So okay, let me explain the more detail of my data. So first one is the untreated leaf data. In this study, we collect about 500 leaves of the five species in our campus. Then I just put the leaf material on this light table, then capturing the image data by digital camera with the specific frame, which have some markers. We can collect the distortion based on these marker information. And if you need, we can introduce some size information by using these kind of the markers. And another one is the cleared leaf data, cleared leaf image data. So we use this open public data set, and we make several types of the subset of this cleared leaf image data. One, the full quality image data set. It is used for the training the leaf image quality recognition model. So then, we also make a high quality data set. It's used for the training the segmentation model. So, and finally, we also make a cleared leaf data, which is consistent of about 300 good quality leaf image data of the 5 genus correspond to these 5 species. So by using this image data, firstly, we make a segmentation model using a unit. So the point is that we use the high quality data for training the unit model for avoiding the annotation cost. So yeah, could you imagine the effort on the manual annotation of the leaf venation pattern? It's one of the nightmares for me. So then we firstly select the good quality leaf venation, cleared leaf image data, then just applying the conventional image processing to make a mask correspond to the original images. Then, based on this training data, we train the unit model. Then, the trained unit model can estimate the segmented leaf vein images from both the untreated and the cleared leaf data. So from the segmented images, we extract the undirected graph representation. So I skipped the detail, but I briefly explain the process. Then firstly, we extract the topological skeleton based on the segmented images, then extract the endpoint and the branching point as a node of this graph. So finally, we connect these nodes with the edges. So we make this undirected graph representation based on the sample data. So finally, we calculate the network-wide scale invariant features called the net CML. So in net CML, the 7 types of the node features are calculated on all of the nodes, right? Then also the summary statistics of these 7 features are calculated. Then finally, we got the 28 dimension vectors as a representation of this kind of the venation pattern. Okay. So that's our proposed method. So let me show you the several examples of each process. So firstly, these are the examples of the segmentation and the undirected graphic representation. So as you see, I'm not sure, resolution is limited, so then, but we successfully extract the leaf vein lesion from the original images, like on both of the untreated and the cleared leaf image data. And based on these segmented images, we also extract the undirected graph like that. If you want to see the details there, please come to me after the presentation. So then, I will show the detailed structure of that. So, and finally, now we can calculate the network features based on the undirected graphs. So I'd like to demonstrate that these network features have enough information to distinguish these species based on the only network features. We don't use any kind of the information about texture or color or something like that. So here we just use the random forest model to recognize to classify these 5 species from the network features. Then in this analysis, the random forest model can classify the 5 species with about 90% accuracy. So it means the network features have enough information for distinguish these 5 species. It's just a demonstration. So then, let's move on to our main result. So that one is the summary of the network features calculating based on the untreated leaf images and the cleared leaf images. So then we just apply the PCA on the network features of the untreated and the treated cleared leaf images separately, right? But interestingly, we can observe some characteristic distribution, one-dimensional distribution pattern in both of them. These, how can I say, the PC space are, reconstruct separately, but we can find basically the same pattern. What is that? So to understand this distribution pattern, we put crop the segmented images instead of these markers, like that. So could you see this region? So then we can find a very interesting pattern. Here, around here, these higher order veins showing how can I say, the open tree-like structure, like that. And in this region, we can also observe the closed loopy structure, like that. So, and the higher order venation pattern are gradually changing along this line from the tree-like structure to the closed loopy structure. But why? So we found a very good previous study. In this study, the authors explored the optimal network structures based under the 3 functional demands. One is the transportation efficiency, and second one is the construction cost of the network, and the third one is the robustness to damage. They try to find the optimal network in these 3 functional space. Then they finally found this red region as a palette front of against these 3 functional demands. So palette front means that a subset of this functional space which cannot improve any functionalities without degrading others, right? It's one of the, these are the palette front. And along this palette front, they also found that these network patterns are changing from the closed loopy structure to the open tree-like structure. Maybe we found that basically the same latent space on venation patterns on the leaves. So these 3 functional demands are basically consistent to the functional demand of the ecological or physiological functional demand of the leaves. So then, we can find basically the same pattern, one-dimensional U-shape distribution pattern, and in this one-dimensional pattern, the veins, higher order veins showing the open and tree-like structure to the closed loopy structure. So maybe the plants or venation patterns occupy this specific subspace of the phenotypic space through as a consequence of the evolutionary process under the several functional demands. Okay, so in this study, we developed a simple and a high throughput phenotyping workflow for quantifying the vein network and identify the leaf venation-specific morphospace. And these patterns maybe correspond to the palette front of the optimize transport efficiency, the construction cost, and the robustness to damage. So I would emphasize that these latent space, we can identify these latent space because we quantify the venation pattern as a network, not just a simple measurement. So I would like to say that, we need to use appropriate model or representation of the target for understanding the phenomena or some structure you're interested in. So thank you. That's all.
Thank you very much, Koji, and I think we have enough time for some questions.
Hi, it was a really interesting work.
Thank you.
So I have a question, because you are using the graph topological structures to do this latent space, but are you using any features? For example, in each node of the graph, you may have an X and Y position that will tell how are these nodes positioned inside the leaf, or are you using only the topological general information?
Yeah, I think we can use such a kind of information in addition to this purely just only on connection information. But in this study, we just use this network undirected graphs representation, excluding the other type of geometric features like length, width, or angles, or something like that. But I think we can add such a kind of information into our analysis.
Thank you.
Further questions? If not, then I have a question.
Oh, sorry.
Please.
Oh, yeah, sure.
Okay.
Yeah.
In the graph that you showed for the PCA,
Yeah.
first two axes, there's a in the central lower graph, there's a strong horseshoe kind of pattern that you can see. Have you checked maybe using a detrended principal component analysis if this is an artifact, or is that really derived from the data?
So you mean what kind of the artifact?
Yeah, typically when you see in PCAs these kinds of horseshoe bow effect, yeah, that could hint at the data maybe not being optimally suited for PCA.
Thank you. It's a very interesting point. And we check, not check the artifact or not, but we also try to find out which network features correspond to the PC1 or 2 or 3. So then we found that the PC1 and the 3 space correspond to the kind of the, how can I say, the number of the neighbors of the AgoNet. It is one of the one-hop subgraph. So then it means that it can capture such a kind of the closed loop or open structure by this PC space. So and we also found that PC2 is a kind of a noise of the images. So then maybe we can understand and interpret each PC space, PC axis related to what kind of the network structures. So then I believe that this structure is not artifact on that. Yes, thank you.
Thank you.
And if I had time, so just I'd like to advertise our other work. So we developed several types of the method to quantify the very diverse morphological properties. So today I talked about the leaf venation pattern, but we also try to develop a method to reconstruct the 3D leaf edges and the 3D surface structure of the potato. But maybe this approach can be applied to any kind of the 3D surface structure, like a fruit shape or something like that. So we also try to optimize the process for getting the 3D information. So then, some members come here and provide a poster. So that please, discuss with them. So thank you.